Good evening, everyone. Biochemistry Simplified. Madam Winnie here. Today we are going to introduce the topic disaccharides. And from the name, disaccharides involves two sugars, two monosaccharide components to be specific, which are going to form the disaccharide. So di means two, meaning two of the monosaccharides involved in bonding. At this point, we are going to talk about the disaccharides. A glycosidic bond is formed. Glycosidic bond is for the carbohydrates. So at that point, a glycosidic bond is forming. There is a release of water. So I mentioned in the monosaccharide the cyclic form. Very important. We drew our monosaccharides in linear, which is Fisher's projection, which made us to understand how to draw the cyclic or Haworth projection. So we are drawing the Haworth projection because at the point we are going to draw our disaccharides, the disaccharides have to exist in cyclic form. So we talked about the cyclic form, example being alpha-D-glucose, Beta-D-glucose, alpha-D-fructose, Beta-D-fructose, or even alpha-D-mannose, Beta-D-mannose, Alpha-D-galactose, Beta-D-galactose. So if we talk about alpha and beta, I mentioned in the previous series of the monosaccharides that we have to draw in a cyclic form. Now, the drawing of those monosaccharides in cyclic form will be helpful to understand disaccharides. So the disaccharides have to exist in cyclic form because in their cyclic form, it increases their solubility. Solubility is increased if the disaccharide exists in a cyclic form. For example, we are going to look at three of the disaccharide. The first example we might be talking about, or we have to talk about, is maltose. Maltose, for example, I don't take beer, but maltose is a component of the malta, which is used in preparation of the beer. For the people who take maltose or the malta, they will always tell the bartender, bring more and more. The solubility, it is sweet in their mouth until they keep on ordering more of the bottles. So we are talking about increase in solubility when the disaccharide exists in cyclic form. Another example is lactose, the milk sugar. We are talking about fresh milk. Fresh milk, we use it to prepare tea in the morning. It has to link well with water. We are talking about increasing the solubility. When we talk about disaccharides existing in cyclic form, solubility is increased. So the lactose, the milk sugar, an example of disaccharide, it links well with water to prepare tea in the morning. The last example being sucrose, the table sugar, which we use to put in our tea. Increase in solubility. I'm insisting on increase in solubility if they're in Haworth projection or cyclic form. That sucrose, if you stir it even in cold water, it is going to dissolve. Increase in solubility. So the disaccharides have to exist in cyclic form to increase their solubility. So we are going to look at the three structures of the three disaccharides and the monosaccharide component which are involved in the formation of the glycosidic bond or in the formation of the disaccharides. And we are also going to stress much on the component, whether they are reducing or it is one of the non-reducing sugars. Why is it reducing sugar? Structurally, if you look at it, how you can tell if it is a reducing or a non-reducing sugar. So let's proceed and look at each one of the disaccharides. Maltose. I've said is the malt sugar. Malt is used in preparation of the beer. It is formed by linking the first glucose you see here, the first one. If you count the cyclic form, remember we mentioned that glucose being an aldose, in cyclic form it is going to form a 6-membered ring called a pyranose, but there is a hemiacetal because of the OH at the carbon atom number 5 forming a covalent bond with carbon atom number 1. So the 6-membered ring tells us much, that it is in Haworth projection or a cyclic form. But we said for the alpha, the OH is below. So the first monosaccharide component is alpha-D-glucose because the OH is below. The second monosaccharide component is a beta-D-glucose. Now, we are talking about specific monosaccharide components for each of those disaccharides we are going to discuss. So if I talk about beta, you can see the OH is above at carbon atom number 1 because carbon atom number 1 was the carbon which was determining whether it's alpha or beta. But the linkage is between carbon atom number 1 of the first glucose residue, which is an alpha-D-glucose. It is linked to carbon atom number 4 of the second glucose residue, which is a beta-D-glucose. So they talk about alpha 1, 4. It means the alpha comes from, it is determined by the first monosaccharide component, which is involved in the formation of the structure of the disaccharide. So in this case, we have an alpha-D. It will give us the name alpha. It is at carbon atom number 1 where the first linkage is, and it attaches itself to the 4th carbon atom. So they talk about alpha 1,4 glycosidic linkage. Then it's also mentioned, or when you do the tests by using the Benedict reagent. This test we could even do it in secondary school, sugar tests. You will find that reducing sugar, if you give it time, it will change to brick red. Sometimes the students will talk about orange color. That is when it changes because the Benedict solution is blue in color. The component of copper sulfate brings the blue color. So by the time it changes the color, you talk about a copper oxide, and already we talk about our sugar being a reducing sugar. So maltose is a reducing sugar. If you look at the second monosaccharide component, which is the beta-D-glucose, there is an OH there. The presence of the OH gives it a property of being a reducing sugar. Lactose. I said this is milk sugar. The component in lactose is a beta-D-galactose and beta-D-glucose. So the first monosaccharide component has to be a beta-D-galactose. Then the second one has to be a beta-D-glucose. If it is not beta-D-galactose as the first monosaccharide component, then the structure is wrong. The first one has to be a beta-D-galactose. And if you look at the structure of galactose, we talked about galactose and glucose being epimers at carbon atom number 4. So you'll find at carbon atom number 4 and number 3, the OHs are on the same direction or at the same side. So they lie on the same side. So the first one is a beta-D-galactose. The OH is above at carbon atom number 1. The second one you can see here clearly, it is a beta-D-glucose. The OH is above. So since we talk about the linkage is beta-1,4, it means being the first monosaccharide component being a beta-D-galactose, the OH is above, it's easier to extend the glycosidic bond up. So it forms a beta-1,4. At carbon atom number 1 of the Beta-D-galactose, carbon atom number 4 of Beta-D-glucose, we are going to form beta-1,4 glycosidic bond. But in between, when the glycosidic bond is forming, there is a molecule of water being released. So we talk about formation of a beta-1,4 glycosidic bond. So most enzymes, in most cases with people who have, rare cases where people have lactose intolerance, it means they can't digest lactose. Definitely we talk about the lactase as an enzyme in the small intestine is missing or is defective. So they can't break down lactose to the simple component, monosaccharide component, the Beta-D-galactose and Beta-D-glucose. So for those people who have lactose intolerance, mostly they have problem whenever they take anything which has milk in it. Because of the absence of the lactase, it can't be broken down into those simpler monosaccharide components. Another point which I want to mention before I can move to the next one is even lactose is a reducing sugar. If you look at the lactose itself, the second monosaccharide component, which is a Beta-D-glucose, has the OH present. So that makes lactose also being a reducing sugar and is be able to reduce the Benedict solution. Sucrose, the last one, is the table sugar. It's also present as a cane sugar. It's the one which is always linked to dental caries. When you have a problem with the teeth, they always talk much about dental caries. So it's related that. But then it is a water-soluble plant carbohydrate and also transported easily in the plant circulatory system. But sucrose is involving alpha-D-glucose as the first one, monosaccharide component, and secondly we have a beta-D-fructose. Still we are talking about the cyclic form. An alpha-D-glucose. Alpha-D-glucose, the OH is below at carbon atom number 1 as much as you can see it, and then for the beta you can see the OH is above. So when the connection is done, we have the alpha 1, 2. The alpha represent much of the position of the OH at carbon atom number 1 of alpha-D-glucose. Then we have the OH above at carbon atom number 1 for the beta-D-fructose. So it will easily be extended downwards because the OH at carbon atom number 1 is below the ring. We are going to form an alpha 1, 2. 1 is the carbon atom number 1 of alpha-D-glucose, and then 2, that is where the confusion comes in for most of the students. Madam, why is it 2 while it's not supposed to be 5 or it's not supposed to be 1? So I take 2 as the second carbon which has the keto group for the fructose. So we have alpha 1, 2 glycosidic linkage forming. But now if you look at sucrose, the second monosaccharide component had been involved in bonding. So it is not as free. We don't have the free acetyl group as seen in lactose and maltose. So all the monosaccharide component has been involved in bonding. So that one makes it a non-reducing sugar. So in this case you can see it has been involved in the formation of the bond. So in this case sucrose is a non-reducing sugar. It means it cannot change the color of the Benedict reagent. It will not be able to change. It will retain its color as blue.